So it's a pleasure to have here Tommaso Giovannini from Tor Vergata University. I just got a tenure track position, became an associate professor soon. So yeah, today we present multiscale approach for computational spectroscopy of complex systems. Okay, it's up to you.
Okay, thank you, Claudio, for the nice introduction, and also to Arian for inviting me to give this seminar. So today I will speak about the developments of multiscale approaches for computational spectroscopy of complex systems that we have done, let's say, in the last 10 years, more or less. So with complex system, what I mean is various chemical and physical systems that can range from a solvated system that can be complex system, since you have a solute that is embedded in an external environment that's generally homogeneous and isotropic. You have a system, let's say a molecule, a chromophore that is embedded in an external environment, for instance, a biological matrix that can be heterogeneous and anisotropic. And then you can have systems where you have composite parts with different macromolecular characteristics. For instance, you can imagine a surfaces with a chromophore embedded in a biomolecule, all surrounded by a solvent. So in these cases, the problem is that you want to describe the electronic structure of these systems. And to do that, you generally need to describe a very large number of degrees of freedom from both the electronic and the nuclear part. And this is something that we actually do not need to treat all the properties of these systems, especially when you are focusing on just the property of the molecule. What I mean with that? So these are some of the most used examples that are taken from the literature. So if you take, for instance, this molecule that is NIRED, a chromophore, or beta-in-30, that is also called the Reichert dye, since it was studied a lot by Professor Reichert, you can see that if you change the solvent, so the external environment, the properties from absorption, fluorescence, and in the case of beta, and I just reported the absorption, you see that it changes drastically. For beta, and it can go from transparent to red to violet to green to blue. Okay, so it can varies a lot depending on just the environment. When you take beta-in-30, it is just a violet black powder. Okay, so this is for optical properties, but this is not only limited to optical properties. If you take a molecule like doxorubicin, that is an anti-cancer drug, this molecule intercalates into the DNA. So it is in particular relevant to understand whether this is intercalated, and so it is actually active from the anti-cancer point of view, or whether it is dissolving the environment, that is water for biological systems. And here you see that, for instance, if you study the resonance Raman, if you measure it in DNA or in water, they drastically change. Okay, so it can be a measure of whether the environment affects the spectral signal, and so when it is intercalated or not into the DNA. But here the important thing is that you are looking at the normal modes and the Raman activity of the molecule as affected by the environment. Another example that as a chemist we like a lot, this is methyloxylane. So this is one of the two enantiomers that methyloxylane has. This is the R enantiomer. And then if you measure the optical rotation, that is the degree of rotation of the linearly polarized external radiation that you are sending to the sample, you see that by only changing the solvent, you can change this angle, okay? And this is particularly relevant because one angle corresponds to one enantiomer. Okay, so if you change the angle, then you need to be able to predict that change due to the solvent. Okay, because of course the only chiral thing that is inside the solution is actually the solute in this case. Okay, I think that all this example can play a role in understanding why, if you take a molecule, a chromophore, and you embedded it in an external environment, the property still comes from the chromophore. In this case, the property comes from the beta-in-30. So we are always looking at the transition of beta-in-30. Okay. But what is important is that the environment affects but not determines the property of the solute. Of course, it affects it, so it perturbs the property, the electronic structure, but we can focus our attention on the molecule. And this idea is actually the idea behind the development of multiscale models, for which Karplus, Levitt, and Worshell won the Nobel Prize in 2013, and can be applied to a very large variety of systems. And it's not only focused on chemical, physical systems, but to also surfaces, composite materials, and so on. Okay, the idea behind the multiscale models, in particular, if you want to do spectroscopy, so the spectroscopic signals are property that arise from the quantum world. So at least the target part of your system, the one on which you are focusing your attention, will be described at the quantum mechanical level. And you can choose to use the method that you prefer: density functional theory, tie binding, highly correlated method, coupled cluster, and so on. While the environment, since it affects but not determines, so you're not actually interested in describing the electronic properties on the environment, you can treat them using a less sophisticated approach. And then you translate your problem into a proper description of the target environment interaction. So if you take your system and you do this partition, so target environment and then a target environment interaction, your total energy will be written as the sum of the three portions, actually the two portions plus their interaction. And then by using many-body perturbation theory, you can see that the interaction can be decomposed into an electrostatic interaction, a polarization interaction that can be properly defined within a classical physics framework, and then two interactions that arise from the quantum properties of the matter, meaning polyrepulsion and dispersion interaction, that are generally called altogether van der Waals interaction. Okay, now the problem arises on how you describe the environment. So we said that you describe it with a less sophisticated method, but what does it mean? So you can have generally two limiting cases. On the one hand, on the right, you see that you take your target system, in this case this chromophore, and then you describe the environment by means of an homogeneous continuum dielectric. Maybe most of you have known the polarizable continuum model. This is exactly what the polarizable continuum model does. So it describes the environment as a continuum dielectric, and then when you know your permittivity constant, then you know how the environment responds to the quantum mechanical density. On the other hand, you can retain the fully atomistic picture of your environment by using fully atomistic approaches. Which are the advantages and cons of both approaches? On the one hand, when you use the continuum, this is easy, let's say, from a conceptual point of view, because you just need the permittivity of your environment to describe its electric response. But of course, you don't have any atoms in the environment. So specific target environment interactions cannot be described. For instance, if you have hydrogen bonding patterns in your between the target and the environment, this cannot be described by continuum approaches. On the other hand, if you take a fully atomistic perspective, then of course this is much more general. You can describe in principle also the target environment interaction, and generally this should be preferred. And indeed, the methods that retain a fully atomistic picture of the whole system are the so-called quantum mechanics, molecular mechanics, or QMMM methodologies, where your target is described again at the quantum mechanical level, and the environment is described by means of classical molecular mechanics force fields. Okay, and so the energy becomes the energy of your QM part, the energy of the MM, and the interaction energy between the QM and the MM portions. Of course, as I said, the MM part is treated by means of classical force field. This means that generally, when you use QMMM approaches, you focus on determining how electrostatic and polarization affects your electronic structure. Okay, so you limit your attention to electrostatic and polarization. How you do that? There are generally two approaches that are exploited. One are the electrostatic embedding that are very easy. Let's say that you take your QM part your QM portion that is treated at the quantum mechanical level, and this will have a density rho. And then in your environment, you put charges, fixed charges in your environment. And then what you will have that is the interaction energy will be just the Coulomb law between these charges and the quantum mechanical density, written in this way. Of course, this has a contribution to the Hamiltonian, to the Consham Hamiltonian, to the Fock matrix, depending on the method that you are using, okay? Because you just differentiate this expression with respect to the density of the QM, and you will obtain the contribution. In this way, in this case, you are polarizing the QM part, but not vice versa. This is, of course, unphysical, because once the QM density changes, then also the environment should adjust to this variation in the QM density. This mutual polarization is introduced by polarizable embeddings, where you are actually include in your environment also some variables that are polarizable from the QM density. Okay, and so your interaction will become electrostatic and polarization. So it's kind of obvious that electrostatic embedding, that polarizable embedding is much more sophisticated and accurate than electrostatic embedding. So in the last 10 years, we have developed mainly two polarizable approaches, two polarizable QMMM embeddings that are called QM fluctuating charges and QM fluctuating charges and fluctuating dipoles. In the first one, in the fluctuating dipoles charges, you just put a charge on each atom, okay, Q1 and Q2, for instance, for water in this case, and then this charge, differently from electrostatic embedding, will vary as a function of the QM density. So this charge will explicitly depend on the QM density. These are solved by solving a linear a set of linear equations. Then, of course, you have just charges, and then we thought, okay, this charge, by definition, a charge is, of course, completely isotropic. Then if you want to add anisotropic interaction in your system, then you can continue, instead of just putting charges that depends on the QM density, maybe we can also add a dipole moment that will respond to the QM density. And then with this second approach, we call the fluctuating charges and fluctuating dipoles. And the interaction energy in this case become the charges multiplied by the potential of the QM density, exactly as for QMFQ, minus the dipole moments interacting with the electric field produced by the QM density. And these charges and dipole are obtained by solving a linear equation, a set of linear equations, okay? That depends explicitly on the QM density. In this way, we are introducing the mutual polarization because, of course, then these charges enters into the Hamiltonian and dipoles entering into the Hamiltonian. Okay, once you have that, then you will have the energy. When you have defined everything, you will have the energy. Of course, in order to do spectroscopy, you need to propagate what you have into the Hamiltonian towards energy derivatives, linear response equations, and the property that you would like to describe. For instance, here I just reported the equations to obtain the optical response of a molecule embedded in a polarizable embedding. This is time-dependent density functional theory equation. This is the Cassidy equations. You have A and B matrix that are modified. Why they are modified? Because once the transition happens in your QM region, you need that the external environment linearly adjusts to this variation. Okay, so the polarization also has an effect also on the excitation energy, a direct effect, not only the modification of the orbitals, but also a direct modification of A and B matrix. Then this is a time-dependent density functional theory. You can do infrared spectra. This is just an example by differentiating the energy 2 times with respect to 2 external parameters that can be the field and the position to calculate the dipole moment with respect to the normal modes. It can be 2 times the position to calculate the action and so on. To calculate, you can also add and go towards the third derivatives, analytical expression. This means that will require, of course, to differentiate 2 times with respect to the electric field and 1 time with respect to the Cartesian coordinates. Indeed, in the last years, we have done a lot of developments in this context. We have a lot of spectroscopy for which we have an analytical evaluation of the response in these methodologies in a framework of density functional theory description of the QM part. So you can describe UVVs, infrared, Raman, as I said before, but you can also calculate chiroptical properties like optical rotation, electronic circular dichroism, vibrational circular dichroism, Raman optical activity, and so on. If you want to have a look to actually what we can do with these methodologies, you can reach our recent reviews in these topics. The methods are also available on Amsterdam Density Functional, so the Amsterdam Modeling Suite, and also in ET, that is an open source code. Okay, and then now I will show you how you actually calculate a property by using these methodologies. So imagine that you start and you have a fully atomistic description of a system. So you have just one snapshot of what is happening in your solution. And this, of course, corresponds to a specific point of the phase space. This, of course, is something that is not reasonable to describe a general spectrum that is modeled and measured from the experimental point of view. And in fact, what you need to do is that you need to sample the phase space in order to have a reliable description of what is happening in your solution, for instance, in your complex system in general. So we have developed this protocol that involves basically 4 steps. You start by running a molecular dynamics simulation. You extract from the dynamical simulation after you analyze them in terms of the hydrogen bonding patterns, the radial distribution function, and so on. You extract a set of representative snapshots, and on each of the snapshots, you run a calculation. And then you will get one of these stick spectrum here, and then you average and you will obtain your spectrum that can be compared with the experiments. Okay, just to show you the quality that you can get with these methodologies, with these polarizable QMMM approaches. I will focus on water as an external environment because water is very difficult to be described with other methodologies since it has specific hydrogen bonding effects. Here it is curcumin, that is a food colorant, and here you see this optical absorption spectrum calculated at the time-dependent density functional theory by means of the continuum approach, so where you don't have any description of hydrogen bonding, or by using our polarizable QMFQ model. And you can see that by comparing with the experiment, the QM PCM is basically wrong of almost 100 nanometer, while QMFQ has an almost perfect reproduction of the experimental band. The same happens for infrared and Raman. This is methyl lactate, again in aqueous solution. For the infrared spectrum, this is the agreement that you can get, and this is also for the Raman spectrum. Notice here that we did not scale any frequency, so this is something that arises genuinely from the experiment, from our calculations, and also the back broadening that is almost perfectly reproduced by our models actually automatically arises from our sampling of the solute-solvent phase space. Finally, a final example, this is N-methylacetamide, that is a building block of proteins, and this is the resonance Raman spectrum, so even more complex spectral signal. This molecule absorbs in the ultraviolet, so in order to measure a resonance Raman spectrum, you need to have a laser source that can reach these ultraviolet frequencies, and for this it was measured by Barbara Rossi at the Electra Synchrotron in Trieste. And again, you can see the agreement that we get with our fully atomistic approach with the experiment with respect to the QM-PCM that is completely wrong and out of scale with respect to the experiment that is normalized with respect to the amide 3 band. Okay. Now we have focused just on the classical interactions. Okay, and now we can ask ourselves, okay, but what about the quantum interaction? We know that this quantum interaction, polyrepulsion and dispersion, can become dominant for many systems, including biological matrices, surfaces, and so on. And the common strategy in a QM classical methodology is to use an empirical potential. Most of you will know the Lennard-Jones potential, right? That has a repulsive part and an attractive part, that is the dispersion interaction. But these corrections that are included corrects the energy because they are parametric in just sigma, epsilon, and the distances between the atoms, but they do not affect the electronic density, okay, of the QM part. So they do not propagate, they do not enter the Hamiltonian and do not propagate to the properties. How you can do that? We have developed an approach that is a general approach for non-electrostatic interaction in QMMM. For the repulsive part, we assume, by following the work by McQueen, that we can write the repulsion interaction as an opposite of an exchange integral. Here you see this rho QM and rho MM are the off-diagonal term of the density matrix of the QM and MM part. This is an exchange integral. And of course, for the MM part, we do not have any way to describe it. Okay, so we need to create a model. What we did was to describe and to create this model in terms of S-type Gaussian function. We parameterize with respect to ab initio models, and then we obtain our values for defining this density of the MM part. You can do an approach also for dispersion. What we did and what we proposed was a generalization of the Tkachenko-Scheffler in QMMM, where you have the coefficients C6 that are defined in terms of homonuclear for the QM part and homonuclear for the MM part that are, of course, parameterized. And then this equation explicitly depends on the QM density, so enters into the Hamiltonian and propagate to the molecular properties. For instance, this is an example of the agreement that you can get with experiments. This is pyridine, again in aqueous solution, and we are studying the M-pi-star transition, moving from the gas phase to water solution. Of course, we run the molecular dynamics, we extract the snapshot, we calculate the absorption spectrum, and we calculate the energy of this transition. And here you can see that the experiment is almost minus 0.3 electron volt. And you can see that if you run just the polarizable approaches, you get something that is more or less minus 0.5 electron volt. So very wrong with respect to the experiment. But as soon as you add the repulsion and also the dispersion, what happens is that you go at minus 0.3 electron volt. Okay, so it actually goes towards the experimental value. So these contributions are actually crucial to go towards the experimental value. And if you get a very good agreement, for instance, in case of an electrostatic embedding where you do not have a polarization, this can be due to error cancellation. You do not have a polarization, you don't have non-electrostatics, and as you get them, then the agreement is worsened. Okay, but these methodologies, of course, depend on parameters. Can we do something that do not depend on parameters to describe the purely quantum mechanical interaction between the target and the environment? Yes, there have been the proposal of quantum embedding methodologies where the whole system is described quantum mechanically. Okay, so in this case, you describe everything quantum mechanically, but the environment, again, they are focus models, they are treated at a lower QM level. What does it mean? I will show you for a specific approach that we have developed, that is multilevel density functional theory, where the starting point is the DFT energy written in terms of the density matrix. Okay, this density matrix, you probably write standard DFT energy in terms of the density function, but these are related. These are the atomic orbitals, okay, and d mu nu is nothing less the coefficient for the expansion in the basis of the atomic orbitals. And this is the so-called density matrix. And then the DFT energy can be written in terms of one electron, Coulomb term, DFT exchange and correlation energies, and if you are in a global hybrid framework, you can have also Hartree-Fock exchange. Okay, the idea of multilevel DFT is that you take your system, even covalently or non-covalently bonded, and then you define an active and an inactive part, and you do a decomposition at the level of the density matrix. So your total density will be decomposed into a density A plus density B, where A is the active and B is the inactive. This, of course, from the definition that I showed you before, automatically implies that the density function is also decomposed. Okay, you take this expression, you plug You do some algebra, you plug into the energy that you had before. This is a bit it's generally. Can you hear me? Yes.
Yes.
You
We can hear.
Yes.
Yeah.
Okay. Yeah. Sorry. Okay. Now, what I was meaning is that you have this expression where you can recognize the active energy, the inactive energy, and the interaction energy. Where you have on the active energy just the energy, the terms that involves A, in the inactive the terms that involves B, and then in the interaction energy you have the mixed term. Finally, you have this term that is due to the fact that these guys, the density, the exchange and correlation density function are not linear in the densities. And so you have some term that arises because of the nonlinearity. Okay, once you have them, you save time in this multi-level DFT because you optimize the density of the active in the field of the inactive, which is instead kept frozen once you have determined it. Then you can write down your Fock matrix, your Kohn-Sham Hamiltonian, and so on, and then you will solve the root annual equation by using this Fock. Okay. In this case, in these models, differently from before, what do you have? You have electrostatics. So this is exactly as a standard QMMM, a polarizable QMMM. But you also have automatically a description of Pauli repulsion, okay? Because the orbitals of the active and inactive are orthogonal. And then you are also including a part of correlation between the active and inactive energies. Of course, what do you miss are dispersion interaction. This is kind of obvious because in DFT you do not have any description of dispersion interaction, you need to include them. Okay, the agreement between multi-level DFT and full DFT can be shown, for instance, by studying interaction energies. In this case, we studied Lysidol in aqueous solution. We took 7 conformers from a molecular dynamics simulation. We took 50 waters around, and then we studied the total energy minus Lysidol minus water. Okay, and then here we reported the correlation between multi-level DFT energies and DFT energies. And you can see that the agreement is almost perfect for all the conformers. And the average error is less than 1 kcal per mole. But you have a computational saving that is almost 70%. So a huge computational saving. You can increase further the interactions, the size of the system that you can use by developing a 3-level approach where you have the active, the inactive, all surrounded by MM. Why you can do that? This is physically grounded because what you can include by adding an external MM layer is long-range interactions that in multi-level DFT are basically electrostatic and polarization that can be properly described by a classical physics. So we have developed this 3-level approach, 3-layer approach, multi-level DFT / fluctuating charges. Everything is polarizable in this case. And then this is particularly useful because now you can focus on the active part and you can run time-dependent density functional theory just on the small part in the active MO space. You do that, of course, you need to modify again the Casside equations where these matrices are defined just in the active molecular orbital space, so they are smaller with respect to the full DFT space. And then you add, of course, the fluctuating charges contribution again, because these charges will respond to the transition density. This is an example, is quinolinium betaine dissolved in various solvent. This is particularly interesting because it changes colors by changing the solvent. So it is the perfect system to see whether our approach actually works and can produce this solvatochromic shift due to the solvent exactly. So we run a molecular dynamics simulation in various solvents ranging from tetrahydrofuran, dioxane, acetonitrile, methanol, and water that are put here in from the lowest polar to the highest polar in this scale that has been proposed by Meyer and al. And then we run multi-level DFT / FQ calculation. Okay, so the multi-level DFT is the region around quinolinium betaine, and then everything, all the rest, is treated by using fluctuating charges. And then we computed the absorption energy. And from the absorption energy, we can compute the solvatochromic shift. And here it is the solvatochromic shift with respect to tetrahydrofuran, so dioxane minus tetrahydrofuran, acetonitrile, and so on. Everything is referred to tetrahydrofuran. And you can see that if you run in red, you have the results by using polarizable force field. If you run the approach that is a quantum embedding approach slash polarizable embedding, what you can get is that in all cases, especially for the most protic and polar solvent, you have that the solvatochromic is reduced and actually goes towards the experimental value that is reported in orange. Okay, so to conclude this part, all these methodologies have been implemented in this open source code. There will be available in the next major release expected at the beginning of 2025. So if you want, you can download it. And then we are now working and developing approaches to describe novel class of solvents. These all the results that I showed you are run for water and conventional solvents, but of course they are associated with an environment impact that can be reduced, for instance, by using deep detected solvents that are novel kind of solvents where hydrogen bonding patterns are particularly relevant and for which quantum effects and fully atomistic approaches are relevant. So I will have 2 postdoc positions opening soon dealing with this problem. So just to conclude the last 5 minutes, until now, I show you just this embedded system where we had a molecule in external environment, mainly solvents. So we have also applied to protein, but I showed you mainly protein, mainly solvents. Can we go and treat also molecules interacting with plasmonic surfaces? So surface where a plasmonic excitation is happening. So if you take a nanoparticle and you put an external radiation, you can excite the localized surface plasmon that are not on the collective excitation of conducting electrons. These are very interesting because you can tune the plasma resonance frequency of the particular for noble metal nanoparticles in the whole visible range by changing the size, the shape, the chemical composition of your nanoparticles. You can go also in the infrared and terahertz if you use, for instance, 2D materials like graphene. And if you put a molecule in the proximity of the surface of these materials, what happens is that you can enhance of several order of magnitudes the spectroscopic signal, and in general, you can modify the chemistry of molecules interacting with these plasmons. You probably know surface enhanced Raman scattering, for which you have an enhancement of 10 to the power of 10. You can even reach 10 to the power of 15 for the Raman signal. Okay. Also, this system can be described by using multiscale methodologies. So this is the power of multiscale methodologies because they can describe also very different systems as the one that I showed you before. Because also in this case, you have the minimal modeling is to describe the molecule, the nanoparticle, and the light, and all the interaction between the 3 players. But what differentiates these measurements, this spectroscopic signal, and this chemistry that happens near nanoparticles and plasmons is that the substrate and the light strongly interact. Actually, you create an electronic excitation, a plasmonic excitation of the nanostructure that can enhance of several order of magnitude the electric field in the surface proximity, modifying the electronic structure of the molecule near the surface. And so to describe this kind of phenomenon, you need to develop force field. If you want to do it in a classical vision, you need to describe force field that are able to respond to the external radiation. We call them omega MM approaches, okay? Omega molecular mechanics force field. How they work? They work pretty well. This is, for instance, gold nanostructure as a function of the size. So this is an icosahedral. You increase the size, and here are our results, omega FQ, F mu. And this is TDDFT results reported in the literature. And you can see by that by increasing the size of the nanoparticle, you are actually getting a very good reproduction of TD-DFT results, even below the quantum size limit. So these nanoparticles are very, very small. They are below 5 nanometers of size. This is actually something that is not obvious. This is actually questions whether we actually need the quantum mechanics to describe the plasmonic properties of metal nanostructures. Okay, by using these models, then you can couple with a molecule. And basically what you need to do is that you need to modify again the response equation of the molecule by including the effect of the plasmon in this case, by modifying the Cassini equations, in this case, in the form of complex couple perturb equations, in order to obtain the perturbed density, and then finally, for instance, a Raman signal as a differentiation with respect to the normal mode of the polarizability. And then here it is just a first glimpse into the experiments of a pyridine over adsorbed on an icosahedral silver nanoparticle. This was the first system on which surface enhanced Raman scattering was measured. And here are our results as compared to the experiment. The agreement is not as good as the one that I showed you before, but the main features that are reported in the experiments are also present in our simulation. And this is a very ideal simulation. We are just putting the pyridine on 3 positions. We should run molecular dynamics simulation to sample the preferred configuration. We should add the effect of the surfactants, the effect of the solvents, and many other effects that are now discarded. Nevertheless, the agreement is satisfactory. So with that, I finished. I would like to thank all the people that worked with me in these developments in the last years, from the group in Pisa, Norwegian University of Science and Technology in Trondheim, the group in Padova, my current group at the University of Rome Tor Vergata, and all of you for your kind attention. Thank you very much.
Thank you, Tommaso, for this talk. There are already there are already different questions. I will start from, okay, keep your hand if you want, from Edward Baxter Linscott. Yeah,
sorry.
Yeah.
Sorry, I was just clapping, not a question.
Ah, okay.
Thank you, Tommaso.
Thank you very much.
I can start with the question in this case.
Okay.
Okay, but just okay. You said it. Okay, you don't have van der Waals because it is not in DFT, but in principle there are some people that developed the van der Waals function. May you think about to extend your approach to this kind of functions?
Yeah. This is something that I'm actually working on right now. So if you take a van der Waals functional, you can plug in into the multilevel DFT definition, for instance, and then you can have part of the dispersion interaction between active and inactive, for sure. Of course, then the question is how good they will be if I took one part of the system as frozen, right? Because dispersion arises because the two parts has a simultaneous excitation in the two parts, right? But this is something I just implemented them in the ET code, so I need to try and see what happens. And the accuracy that one can get within these multi-level methodologies, or if one can try to, let's say, increase the accuracy or define maybe some specific functional to include this dispersion interaction within the multi-level DFT framework. Other ideas instead, for instance, the Grimme corrections are just parametric again. So they will be exactly the same in multi-level DFT as in full DFT, right? So they will work for sure, let's say, because they just corrected the energy, nevertheless, right? So you will have energy corrected, but not propagated to the properties, right? So you will not have a direct contribution into, for instance, time-dependent DFT equations.
Because I guess, for example, I completely agree.
Yeah.
Other question? Otherwise, I ask another one. Okay, I will ask you. The code that you just, because you mentioned the code, the ET.
Yeah, ET, yes.
ET, so is like a post-processing code or is a DFT code from scratch? So
so that code is a code that it started as a couple cluster code from which it comes this ET name, that is the instance that you use in couple cluster on the reference wave function. So it is basically it is an ab initio code, an electronic structure code where we have implemented from scratch DFT. So we implemented a grid. Yeah, the DFT is implemented. The DFT functional are implemented with an interface with libxc. That is the one that most code are actually using for DFT. And then, so yeah, it is from scratch. So it's not a post-processing, but it's an ab initio code, let's say.
Okay. With post-processing, would mean that you read wave function from somewhere. No, so you produce everything. Okay.
Yeah, you produce everything. Yeah, of course. Then you need to do some post-processing because for each of the QMMM or multi-level DFT calculation, you will have one just snapshot. Then you will have hundreds. For some cases, thousands of this snapshot, and then you need to do post-processing analysis of these results, right, to obtain the final spectrum, let's say. But this can be done with a Python script very easily.
I have a small question. Hello, Tommaso. Nice to meet you.
Nice to meet you too.
It's just a curiosity. Actually, at slide 11, you said that there is an active and an inactive part of your molecule.
Okay,
I didn't get the...
go there. Okay, see, yeah, 11 are a bit, okay, here, for instance.
Here, for example, yeah.
Yes. Okay.
How do you maybe I lost a part of your talk, I don't know, but how do you say that one part is inactive and the other is active? Inactive with respect to what?
Okay, so the idea of this method is this one. So you start from let's say a gas density for your system. This gas density can be obtained whenever you want. So for instance, with a superposition of atomic densities for each of the atoms, this is a way. Okay, you get this density. Then you decompose this density in an active part and an inactive part. With active and inactive, what I mean is that at the end, what you would like to do is that you will like to optimize the density of the active in the field of the inactive. Okay, so what you're actually want to do at the end is this thing here. So you will take the density of the active, you will have a dA and dB. The dB remains frozen. So it will introduce a field on your density of the active part of your system that you have defined. And then you optimize this density in this field. Okay. So the idea is that once you have determined dA and dB, you construct this energy and the associated, let's say, constant operator. And then you just optimize this dA in this energetic contributions here. So only dA will vary in your circumsystem field procedure that you will do. So this means, for instance, if you have a solution, imagine that you have a system like this, okay, or better a system like this, where you are defining as red your active part, that is your solute, and with blue you are describing your inactive part. Okay, and then what you want to do is that you want to define and you want to have a look and to determine the best density of your active part, that is your solute, the target system of your interest, in the field of the inactive. The idea is exactly the same as QMMM, but here instead of QMMM, you have a density determined at the DFT level to describe the environment.
Okay.
Did I explain?
The point is, for me, is that I thought that active means that the active part is the part where some physical process that you are interested in resides.
Yes, exactly. So for instance, once, so of course, how you define the active and inactive is up to you, right? So if, for instance, you are interested in the properties of this part here, then that will be your active. In the application that I showed you, so for instance, in this quinolinium beta that is here in green, quinolinium is the active part. Because I am interested in the electronic excitation of this part.
Okay.
Then I have the closest solvent molecules that are inactive, and all the rest that are MM. Okay. Because
okay. Now it's clear.
Yeah.
Yes.
So the idea is that you will calculate the properties just on the active.
Yeah.
Okay, so for instance, independent DFT, you will run only on the active.
Yes.
So the part of the system that you are mostly interested in. Yeah, you are right. Exactly.
Okay, thank you.
Thank you.
I have a question.
Yes.
Also, Sam.
You're muted, Claudio.
Sorry, we have a couple of questions from Asim and Lorenzo. Let's start.
I think Asim was before me. I think.
Yeah, Asim, let's start.
Okay.
We don't hear you. At least I don't hear you.
Yeah.
Yeah.
No.
Can you hear me now?
Yes.
Yes.
Okay. So I have one question regarding the same separation of active and inactive part. So if you're considering electronic excitation in active part, while you can separate, let's say, the ground state density, once you excite the active part, how can you know what will be the effect on the inactive part of this excitation? And then later you will also the change in the charge density distribution in the inactive part, or whether there is a charge transfer excitation, or let's say a mixed excitation, where you have some localization on the active part, but some localization of the electron on the inactive part. So if you just exclude it, then you kind of let's say the transition basis is restricted to active part. So is that justified?
Yeah, so you are completely right. So the idea behind every focused approach is that this is valid for both multi-level DFT, but also for QMMM, QMPCM, if you have run some QMPCM, is that the basic idea is that your property belongs to your active part. That is your QM, your active multi-level DFT, or whatever you want. So as soon as you focus only on that one, you are disregarding everything else. So for instance, you are disregarding the electronic transition that goes from the active to the inactive. These are for sure not included in the modeling, okay? Because the inactive part is kept frozen. So for instance, there are some transitions that are very interesting for where you have a solute-to-solvent charge transfer excitations. This requires a full quantum mechanical description of at least the molecules where the excitation goes. That cannot be described within this focused model idea. Okay, so you are perfectly right. I mean that the models are models so that you need to know which are the things that you cannot describe with them.
Okay. So typically, to exclude such a possibility, one would need to progressively change the size of the QM part to kind of see really what may be happening.
So this is something that one can do. For instance, if you think that you can have some charge transfer excitation to the, for instance, the fast solvation shell, then you can try to include that into the QM region.
Okay.
Okay. So you, or in the active region, let's say. Okay. So in the multi-level DFT, you will expand the active. In a QMMM, you expand the QM.
Okay.
Yeah.
I guess that would also depend on the level alignment between the solute and the solvent, and because it may be possible that even if there is a charge transfer, that happens to be at a very similar energy as the excitations which are localized. So it is just by comparing with experimental absorption spectra, it's difficult to say whether or not there is a charge transfer.
Yes or no. I mean that when you have a when you have a charge transfer to solvent, then you see specific bands that arises, and you are not predicting them with these focus models. So that affects strongly the spectrum.
But how do you a priori know whether this is happening or not?
You do not. You do not know a priori. I mean that for the most large, for the most cases, when you are doing things in solution, for instance, you know that this charge transfer excitation to the solvent are generally very, they are generally absent. But of course, you do not know a priori. I mean, when you want to rationalize an experimental finding, first you try with these focus models. If you cannot predict them, then you may think, okay, where I am wrong, right? So my model is wrong in something, and then I have missing some physical or chemical interaction with the environment, or I missed something in the QM part also. Because, for instance, it can be multi-reference description instead of DFT, no? So, yeah.
Thank you.
Thank you very much.
Lorenzo?
Yeah, thank you. Thank you, Tommaso, for the talk. So I have in mind a similar yet quite different example. I have some experimental friend who, let's say, modifies the emission band of some molecule, not through the solvent, but using encapsulation in nanotubes. Basically, the molecule enters into the nanotube, and depending on the size and the material constituting the nanotube, the emission changes. So I think it's quite similar of the sample, because there is no physical contact between the two regions. So it's quite simple to divide the problem into an active region and an inactive region. The point is that the inactive region is very, very big, or let's say periodic.
Yes.
There is a way for you to include periodic conditions, at least in the inactive part?
Yeah, I mean, this is something I have been thinking about in the last month, let's say. I think that it is something that is doable. I would like to have something exactly as the one that you are speaking about. So only
there may be other
part in the periodic. Yeah, but this is also the case of a molecule into absorb on a surface.
Exactly. This is another
where
example.
you need to have a cell that is large enough not to have interaction with the other cell, right? This is something I've been thinking about. I still don't know how to do, but I will try to do in the future. Another thing that answer to your question. The point for the emission is that so you are going to the excited state and then you relax, right? The geometry in the excited state. And if the emission property depend on the size of the nanotube, it can be that you go to a different geometry in the excited state, and then this modifies your emission.
Okay, right? Yeah, you're right. Maybe this example is too complicated.
No, that's fine. I mean, yeah, the idea is correct.
Idea is but there is also I was thinking also toward another complication, which is the following. So imagine the same system, forget the emission part and the possibility of changing geometry. Just you have this molecule encapsulated into the tube, and the tube has a defect, which is very close to the molecule. And imagine that there is some charge transfer, as you said, to this defect. So if I want to mimic your approach, I would say, okay, there is an active part that is the molecule. The first part, first
Yeah.
shell around, which deserves to be treated in QM, quantum
Yes.
mechanically,
Yes.
but includes only the defect and possibly the surroundings of
The surrounding.
and the rest
Yeah.
of the tube.
Yes.
But there is another point here, is that the division, the separation between the second shell and the mechanical, the classical physics shell is not clear. I mean, there are chemical bonding between.
Yeah, because exactly. So in this case, you are, I mean, you are breaking covalent bonds, and you are actually, I mean, the electrons that you will have will be very delocalized, right? So actually, what you are doing is something that is not very easy to be done in a QMMM, basically impossible, because it's if you want to describe this this delocalization, you need to have the electrons. So in the MM, is something that you cannot do that. In these multi-level methodologies, we have cutted some covalent bonds. The example that I showed on the nicotine is, so I will show again 1 second the screen, just to show you. Do you see the screen?
Actually, the second question can be rephrased in the following way, that is simpler. Is it possible to cut a system into an inactive and an active part? Okay, forget the multi-level. If there are bondings between the two.
Yeah, for instance, this is something, so imagine this molecule. Okay, this is a very simple one. Okay, but this is...
you're right, you cut it,
I cut it, okay, and I have an active and an inactive. Okay, so this is something that in principle you can do. Here I did for a graphene, as very small graphene. Okay, so you see here I cutted all these bonds. Okay, and this is something that in principle you can do. Then of course when you have delocalization, as in this case, then you need to be very careful of what you are doing. Okay, because delocalization properties, you need to properly cut and define the total density before cutting. Okay, but in principle, it's something that this multi-level idea is something that allows you to also cut covalent bonds. Okay, so this is very powerful, more than QMMM. In QMMM, you can cut, and then you need to put link atoms, for instance. This is something that is done when you have biological matrices. But this, of course, cut completely the delocalization. Okay. In this other case, instead, the total density will remain the same. Okay, so you can cut, but you need to pay attention on how you are cutting. But in principle, you can. So of course, depending on the problem, it can be very difficult to do. But in principle, one can try. Yes.
Good. Thank you very much.
Thank you. Thank you.
Okay, thank you, Lorenzo, for the question. So if there are no questions anymore, I can more question. So I thank you again, Tommaso, for the talk.
Thank you very much.
And we'll see you next time in Torre Capital.